Hello, everybody. Uh, we start. I want to thank you to the invited speaker and friend, and also thank you to the general audience. I hope you can hear or listen my voice without problem. I can see the image in your screen. Okay. Well, dear colleagues and friends and general audience, thank you for attending to this meeting, where we share and discuss the investigation focusing how microorganisms are involved in nutrient cycle in aquatic environments, particularly bacterial community and the focusing phosphorus in this time. I really appreciate you stay here connected today, early morning in Chile and USA, and also evening in China, or another part of the world, because we share this invitation for all different collaborators, international collaborators. Okay, this meeting is under financial support of our international cooperation project entitled Research Network for Understanding the Role of the Bacterial Community on Organic Phosphorus Recycling in Anthropogenic-Impacted Aquatic Ecosystem. This project is focused, we try to create a network, international network, with working together in organic phosphorus in aquatic river or lake, very relevant in this area. That's appreciate the cooperation for international collaborator to participate in this big challenge. We is very relevant for our area in Chile, in the south of Chile. Yeah. Also, we invite to Chinese collaborator who are invited like a part another project. Maybe in the future we create another project focus also in the lake. Villarrica Lake is very relevant lake in Chile, and I know you some invited come to Chile. I know Villarrica Lake, and we also can see Marcos Campos. He working in another lake in Chile, also relevant. We focus in not only lake, also river, all different aquatic system in the south of Chile. Very relevant, not only for the people or tourists, also for ecological service system. That's very relevant for us. We just start to investigate that kind of topic here in our laboratory. Okay, this project is financed by Chilean Agency of Science and Technology, ANIT, and in this sense, also in the sense as a part of the Apply Microbial Ecology Laboratory, we expect continue making online meeting in the future, including presentation and international speaker for other projects we have in our other area, like a marine microbiology, like a red tide problem, plant micro interaction, like a plant bureau promoting bacteria, and recently we opened the area of air microbiology, that we had different students and projects in cooperation with international collaborators on bacteria in the air. Okay, please give me, I'll talk a little bit in Spanish for general audience, and they continue speaking in English, okay? Bueno, muchas gracias a los que asisten a esta transmisión. Ya es la primera vez que transmitimos esta modalidad del laboratorio, esta nueva modalidad que tenemos que hacer. Y gracias por su atención y esperamos que puedan disfrutar estas presentaciones. Siéntanse libres de preguntar a través de las plataformas. Nosotros estaremos atentos a sus preguntas hacia nuestros invitados, ya en el área actualmente sobre microbiología en ríos y lagos de acá de la zona sur de Chile, ya. Y esperamos como laboratorio continuar haciendo estas diferentes actividades, diferentes proyectos de nuestra área. Tenemos proyectos tanto en área terrestre como acuática, marina y últimamente en aerobiología, ya, en microorganismos celestes. Así que esperamos que sea de su interés en futuras presentaciones que nosotros estaremos invitando. Okay, I continue in English. Sorry. I would like to thank to our invited speaker. We had in the case Andy, we had a long time cooperation. Thank you, appreciate for your assistance to this meeting. And we start to sharing collaborator. We just start about 1 year ago a new project. That's I sure we have very productive. We find very interesting that. Okay, we can start the meeting. So first, I introduce to Dr. Marco Campo, who helped me in the coordination of this meeting. Please, Marco, go ahead.
Thanks very much, Milko. Good morning to everyone. Buenos días a todos. I hope to be okay in your homes, safe with your family, with your colleagues. I present the first speakers or international invited. That is Andrew Ogram, Dr. Andrew Ogram. Good morning. He's from the University of Florida, United States. He will present the theme entitled Cycling on the Gradient, the Microbial Ecology of the Florida Everglades. Interesting speaker. Dear Andrew, you have 25 minutes for your presentation, 5 minutes for questions. You can start when you want.
Okay, I'm going to try and share my screen now. Let me know if it's working. Huh. Okay. Can you see my slides, Marco? I'm not. I don't know what you can see. There we go. Maybe this will work. Okay. Marco, can you see my slide? Ja oder nein? Okay, great. I'm hoping. All right. So I can't see the chat box, so if you can't see my slide or you can't hear me, please talk. I'll try and speak slowly. It's good to see so many old friends. It's good to see Milko and Marco, and good to see Xiaorong and Deb. More recently, I've seen Deb, I think. So what I'm going to do is talk to you a little bit about not phosphorus per se, but the ecological impacts of phosphorus enrichment in some work that we've been doing over the past 20 years in the Florida Everglades. So I'll give you a brief introduction to the system and then talk about some of the coupled cycles in the Everglades. And those include methanogens and sulfate-reducing bacteria that both compete and cooperate, and methanogenic nitrogen fixation, which is something that we're getting to be very interested in now. So this is the system that we work in. This is the southern part of my home state, Florida. And the greater Everglades ecosystem stretches from really around Orlando in the central part of the state, down the Kissimmee River, which empties into Lake Okeechobee. And this area that the Kissimmee River drains is includes both residential and urban drainage, as well as quite a bit of agricultural drainage into the lake. And the lake is quite polluted through nutrient enrichment, a lot of phosphorus and nitrogen that are going in there. Below Lake Okeechobee is really the part that we're interested in. This is the greater Everglades area. The Everglades at one time was a very large marsh. Over the course of the past 100 years, it's been significant.
Andrew.
Yeah.
Just a second. We are not seeing your slides. Now we are seeing your slides, but you have to put that as a presentation.
It was a presentation.
The bottom, I don't know.
That's his presentation, Marco. So I don't.
Maybe you are sharing your the other screen.
You know, I don't know. I can see. I don't know. I mean,
okay, but we can see the main slides. So okay, you can continue. Sorry.
Okay. All right. You know, maybe this is so. Let's see. All right. So Milko, I'm sorry, this is going to take time. You can see the slide. I'm going to change it from the map of Florida to photographs. Can you see that? Are you seeing the change in slides?
Andrew, I think at the bottom of the screen, you have to please click in the bottom presentation. Here.
You know, it is. I just took it off of the presentation, and so now it's not presentation. Now it is presentation.
Okay, but we cannot see the presentation mode.
Well, I don't know what the problem is.
We continue see you the PowerPoint software, no full screen. But
Yeah,
anyway, you want to try. We continue in this. We can see the slide too. But
okay. Can you see me changing the slides now?
Yes. Now, let's go ahead.
Okay. All right. This is not presentation mode from my screen, but I'll just continue like this then, if you can see it. And so I'm assuming it's very small. I don't know for you all. So that's going to make some of this difficult to see. Anyway, I'm sorry to waste time on this. I'll try and go through this fairly quickly. The primary area that we're interested in is this area just below the Lake Okeechobee. It was drained up about 100 years ago to for muck farms. So sugarcane is the primary crop in this area. It's the Everglades is historically a very, very low phosphorus environment because it's underlain by calcium carbonate from coral reefs, and so that calcium will suck up the any phosphates that's there and results in an extremely phosphorus limited system. And so in order to grow cane, they put on phosphorus fertilizer, as well as in attempt to drop the pH a little bit. Elemental sulfur is also applied, and that elemental sulfur is converted by sulfate or sulfur oxidizing bacteria to sulfate. And so there's kind of a shallow sulfate gradient that goes in as well. This is what it looks like. As you can see, it's very, very flat. It's drained continually by canals, and because of runoff, then you get phosphorus and a little bit of sulfate and nitrogen going into those canals. And then historically, those canals would empty into the northern part of the marsh. And that's been controlled quite a bit recently, but there's historically a high amount of phosphorus that's just below that agricultural area. And it's continually redistributing as the water moves south. The phosphate is also moving south slowly, but we have a gradient in phosphorus concentrations. And so what I'll be talking about primarily today is this area right next to the agricultural area that's a marsh, and the phosphorus rich area, it's been converted to cattail, and the low phosphorus or original marsh, it's low phosphorus, and sawgrass is the dominant vegetation. So you can see by these dark colors the relative concentrations of the phosphorus as we move into the marsh. So we have three basic sites. One is impacted areas that's clearly been impacted by phosphorus. Another that's close to the front of the phosphate moving into the marsh. So this is a transition area. And then a little bit further south is the quote unimpacted area. So what we find is that predictably with increased phosphorus concentrations, you have substantially increased phosphorus or primary productivity. So not only a shift in the dominant vegetation, but also a lot more primary productivity. And that results in increased microbial activity and biomass, and a shift from a phosphorus limitation in that area to a nitrogen limitation for primary productivity and many bacterial or microbial processes. We also see an increase, as you would expect, an increase in peat. in soil development. So there's a lot more carbon coming into the system because of the phosphorus, and so you get a greater rate and a greater depth of peat accumulation. Okay. So one of the things that we're interested in is what that does to the processing of carbon in the along this gradient. You know, as we go from a nitrogen limited system to the more traditionally phosphorus limited system. And so some of the data that I'm going to show you are quite old. This is from the early 2000s. We had ran a geochip, if you recall what that is. Geochip was a microarray that was developed to investigate the response of microbial communities to bioremediation. And so it's an array that has around 27,000 genes, but most of those genes are for aromatic catabolism, aliphatic catabolism, energy generation, things like that. And so it's really kind of a good application when you're looking at biodegradation of peat, of organic carbon. And the thing that's really interesting to us is that or to me anyway, is that in the impacted area, we have around 300 or around 148 unique genes, but quite a bit that's being shared with that transition area. In the unimpacted area, we have almost as many genes that are shared with the transition zone, and a limited amount of unique genes. And so what we see in that transition area is maybe what you would expect. At this point, you see a kind of a mixing of the communities. As you get this phosphorus front going down, you have a mixture of cattail and sawgrass. You know, so you probably expect to have significant overlap of the two end members. But what we weren't expecting is that by far the greater amount of genes that are being picked up by this microarray are unique to the transition area. So it's not just that we're seeing a melding of the communities. We're also seeing this great explosion or great expansion in catabolic diversity in that transition area. So a lot more activity there. I'm not going to talk much about phosphatase or phosphorus, but I do want you to know we've done some work on that in the past. So I think these numbers may be very difficult for you to see, but so if you'll trust me on this, what we're seeing is we look at quantitative PCR of 16S ribosomal RNA genes in the high P area and in the low P area. And what we see is, as you would expect, greater numbers of 16S copies, suggesting or indicating greater numbers of bacteria in the high P area. And then it goes down considerably as we get down to the low P area, as you would expect. You know, a lot more energy coming into that, and carbon coming into that high P area, and really impacting the community. What we see, though, is also, if we look at a couple of different phosphatase genes, PHO-X and PHO-D, two different forms of phosphatase, we see in general, in the high P, we normalize the PHO-D to 16S copies. And so that allows us to kind of get an idea of the general enrichment of phosphatase genes in the community. It's quite low. Those ratios are quite low for most of these systems until we get to a very low phosphorus area where we see several orders of magnitude higher enrichment of phosphatase genes. So I don't think this is earth-shaking, but it's what you would expect, that the greater the phosphorus limitation experienced by the microbial community, the greater amount of phosphatase genes that are present in that community. Okay, we're interested not just in phosphatase, but we're interested in the impacts that this phosphorus has on microbial community functioning and ecosystem processes and functioning. And so we can think about anaerobic decomposition of carbon as being analogous to a metabolic funnel, where it starts at the top with all of this peat and undecomposed material coming in through depolymerization into this metabolic funnel and going through depolymerization, degradation, which includes quite a bit of fermentation, but there are also other processes that are going on. And then finally, out the bottom, we have these mineralization products that are carbon dioxide and methane. And we're also interested in electron flow, and hydrogen gas is an important component of that electron flow. So what I'm going to talk to you a little bit about now is the impacts of this phosphorus enrichment on these fundamental processes controlling mineralization. Okay, this is area 2. At the top, this green part is the phosphorus enriched. Then as we go down into the marsh, it becomes more phosphorus limited. And so that's reflected in the way that methane is produced. Not simply the amounts, but also the pathways that methane is produced. So generally, if you look at the stoichiometry of methane production from cellulose, which is a primary route for it, you get around 67% coming from acetate and 33% coming from hydrogen, reducing carbon dioxide. So there are 2 basic flavors of methanogens: those that use acetate to make methane, and those that use hydrogen and CO2 to make methane. They're physiologically and phylogenetically different. Okay, but there's this general rule of thumb that this is the proportion of contribution of those 2 separate pathways to methane production. And it generally holds. It doesn't hold for northern peatlands. It doesn't hold for some other special cases. What we've found, there are 2 broad groups of sulfate-reducing bacteria. So if you recall, we also have a very shallow gradient in sulfate, because sulfate was generated in these agricultural areas, then it washes off just like the phosphate comes into the system. And so there are 2 broad groups of sulfate-reducing bacteria, and they're also identified by their relationship to acetate. So some can use acetate as a terminal electron, as an electron donor, and some that cannot use acetate. All right, so you've got methanogens that can use acetate, sulfate reducers that can use acetate, and then methanogens that don't use acetate, sulfate reducers that don't use acetate. And so that sets up an interesting competition between these groups for acetate in the phosphorus-enriched area. So this is redundancy analysis, and what we see in this is the number of hydrogenotrophs or hydrogenotrophic methanogens, those guys that do not use acetate, are strongly correlated with general sulfate-reducing bacteria, but they are also opposite to the acetotrophs, or the guys that use acetate. So we thought that was kind of interesting. So methanomicrobiales are the hydrogenotrophs, and then methanosata are the acetotrophs. And so they're completely opposite. They're completely separated. And what we found also is that as a result of this separation, there's a shift in the pathways. We have come to expect that acetate would drive about 70% of the methane produced, 67%, which, we measure 70%, so that's pretty close. But we get a whole lot more methane that comes from hydrogen up in that nutrient impacted area. And that goes along with what I was showing you with this enrichment of hydrogenotrophs. So in that area, which is kind of unexpected. We don't really know what's happening. To make a long story short, I'm not going to show you the data for this, but what we see is a competition for acetate between the sulfate reducers and the methanogens in the nutrient impacted area, and we see a much more standard type pathway down below. All right, so there's this competition that's set up, and the sulfate reducers are able to outcompete the methanogens for the acetate, and that has potentially some significant impacts, not just for the way that methane is produced and the rate at which methane is produced and the potential for response to warming, with climate change, but also some other downstream processes. So one thing is methanogenic nitrogen fixation. Another thing is mercury methylation, and I don't have time to talk about that much, but trying to not to run over. I've got about 4 more minutes left, I think. So what we found is that virtually all of the hydrogenotrophic methanogens in the Everglades have the ability to fix nitrogen and probably are fixing nitrogen actively in all of these environments that we're looking at. So the importance, the ecological importance of this is that down in the peat, everybody is probably limited in nitrogen. And so all along that area, you've got nitrogen limitation. And so the methanogens are making it for themselves. They're making the nitrogen for themselves, and perhaps being an entry point for nitrogen into that peat. So this is a busy graph. I don't have time to show you everything right now, but what I'd like you to focus on is this bar graph right in the upper right-hand corner here that shows the distribution of messenger RNA from nitrogen fixation based on different groups. The blue are bacteria, and the coral colors here are the methanogens. And what you see as you go downstream, you see in the nutrient impacted area, you see a lot of messenger RNA that's controlled by the nitrogen fixing by general bacteria. And then as we get into this further area that's not phosphorus limited, or sorry, that is phosphorus limited, but also has a lot lower carbon input, a lot lower energy input, we see a great contribution to nitrogen fixation by methanogens. And so at least in this area, in the low phosphorus situation, we think that we see a real importance of methanogens in bringing in nitrogen into that peat, which is important for decomposition of the peat, maybe important for decomposition of the peat. So the enrichment, the phosphorus enrichment, has the effect of changing the way that nitrogen enters the system, and also nitrogen limitations. All right, so in summary, see how much time I've got. I've got 1 minute. The nutrient impacted peat versus the low nutrient impacted peat. We see primary productivity is shifted toward a nitrogen limitation. We see expanded catabolic diversity in that area. In a transition zone, we see a greatly expanded catabolic diversity, and the microorganisms are really attacking that stuff. And then there's this potential shift in the way that methane is produced, or there is a shift in the way that methane is produced, and also in the way that nitrogen is fixed in that peat. So I that brings me to 8:30, and so I think I have 5 minutes for questioning. So be happy to try and answer any of the questions that you have. Now, I hope I didn't speak too quickly for you. We have a lot more data on this system, but I tried to kind of limit it, but so there you go.
Hello. Thank you, Andrew. Marco, are you there? We try to collect some questions.
Okay, Deb asks how pH limitation is calculated, and so it's a functional assay. So it's basically.
I have a question here, Andrew.
Yes, we have a question here from Gonzalo Gajardo, Professor Gonzalo Gajardo. He's asking, how do you see this aquatic system evolving in a context of climate change?
Okay, well, that I'm not really sure about, but as you, some of our colleagues that are working in the same system have shown that as you gradually increase temperature in these peats, these same samples, you increase the hydrogenotrophic pathway, and you also change a little bit the way that carbon is processed in those samples. And so you would expect for the hydrogenotrophic pathway to be a little bit more sensitive to temperature because of thermodynamic constraints, but also you see a little bit more increase in decomposition of the peat. So the greater the amount of, the greater the temperature, the greater the enzyme activities, the greater the methane that's produced, and the greater the, and also a shift in the pathway, a continuing shift toward hydrogenotrophic methane. And so the outcome of that is that it is not a simple linear effect. It will probably be a little bit more than additive with climate change. So
okay,
and
there is another question from Deb.
Yep.
Our next speakers. He asking if the analyze were in sediment or waters or both.
Okay, so Deb has actually two questions. One is very good. How do you know it's nitrogen or phosphorus limited? And so those are done by a few different ways. One is by enzyme assays. So you see changing ratios of in-processing and nitrogen processing enzyme activities as a ratio of carbon processing enzymes, and the same for phosphorus processing enzymes to carbon processing enzymes. And so that's one way to do it. The other is just very functional. You know, you put a little bit of nitrogen in it and you see a response, or you don't see a response. So it can be very functional like that. The way that we've done it is largely by enzyme assays, though, that are characteristic of nitrogen and phosphorus limited systems. So Deb asked if these analyses are in sediment or in water, and so these are in soils. So they're in peat. So this is a very complex ecosystem. So you've got water, and there's periphyton, floating periphyton in it, flock, and then you also have peat, and everything I told you about today is in the peat itself.
I have the last question. How you link the methanotroph and hydrogen bacteria and the sulfur reducer bacteria with the phosphomonosterase activity?
So we haven't.
Okay.
That's the answer.
Not directly linked in this.
Not directly. So this is
okay.
Yeah, we have not linked any of that, and I have no idea if the methanogens are producing phosphatase at all. It's just the phosphorus enrichment has changed the environment for the activity of these guys, and so that's it.
Nice. we can remember to the people that the final speakers, we have a round table for more questions. So we want to say thanks you, Andy, for your presentation, your interesting presentation. We will leave time for the next presentation. Bilko, please.
Okay, thank you, Andy, for your nice presentation. Okay, we return again in the end of the seminar. We had a round table. Also, you can remember or remind to the people if they has a question, please. You can write and we can read later when they finish the presentation. Okay, all right. We continue. The next speaker is the Dr. Dave Yacy. We had also very nice collaboration with him from Delaware University. Hello there.
Hello.
How are you?
I'm good, thanks.
Thank you.
Right. How are you?
Thank you for collaborating with us. It's very relevant for us. Appreciate. Okay, we have time for the presentation. Please, 30 minutes in total, and we have 25 minutes to presentation and 5 minutes per question. Also, we try to collect questions for the general audience too, and then inform it to you. Okay, thank you. You can go ahead.
Okay, sounds good. Thank you. All right. Let me share the screen. Okay, does it show in the presentation mode? Hello? Okay. Give me a second. Looks like the presentation still shows similar problem. Okay. So let me talk about the phosphorus sources cycling and touch upon how it impacts the coastal water quality. I have a series of co-authors, some of them my past student and postdoc who work hard in this project, and a researcher from University of Oregon and Pacific Northwest National Laboratory. Let me talk in general about the water quality issues in the Chesapeake Bay. The Chesapeake Bay is the largest and most productive estuary in the US. It carries about $1 trillion economy with about annual benefit of $30 to $60 billion. It's coined as a national treasure by executive order of the past president. That means anything in this watershed has a national interest. The eutrophication, the water quality issue, primarily because of nutrient, goes back to pretty long, about 200 years, where the signs of increased phytoplankton and then gradual decrease in water quality, water clarity, goes back to about 100 years. But since 1950s, and largely from 60s and so on, there are more and more evidences and cases of hypoxia or anoxia and gradually loss of submerged vegetation. This is just a couple of numbers on how much cost we as a taxpayer pay for water quality improvement. Left on the top, I'm showing the 2009 to 12 milestone, how much phosphorus and nitrogen has to be reduced from dominantly agriculture source, about 61 to 63%, to get the water quality to the current state. This number has not changed much until last 2 years. And below is the number, how much every state, watershed states are contributing to meet the water quality goal. The figure on the right, I'm showing how the water quality standard has remained subpar. If 100% being the acceptable or before the eutrophication water quality, we stand about 35 to almost 40% without a significant improvement. So that brings a question on many fronts. And so what is going on? Is it because of some of the basic questions on phosphorus cycling we don't know, or the effect just takes longer time in bigger water set? So our research question in my lab and in collaboration are centered on speciation and characterization of phosphorus. And we use isotope technique to identify sources and pathways of phosphorus cycling. These are a couple of pictures and a cartoon showing the way we collect sediment and water. We use normally UD SIP sharp and collect the sediment core and then process those core for phosphorus extraction. We also collect lots of water from overlying water column and then process them for extraction and finally precipitate as a silver phosphate if the isotope analysis are needed. Let me talk about the water column processes, primarily particulate phosphorus, in terms of question on remineralization or formation of any inorganic mineral from particulate matter sources. On the right, I'm showing Chesapeake Bay water body only, and the color index here are for degree of anoxia, the dissolved oxygen concentration. As you can see, from 0 to 12, 12 being the most toxic in this water, 0, of course, is anoxia. At the central site, as you can see, 3 sites that's being studied in this project has intense anoxia in the water body. On the left, I'm showing very old data on particulate phosphorus. It's shown as PP. As high as 90% of phosphorus could be in particulate matter. As you all know from phosphorus chemistry, it remains as a sorbed species or colloidal species rather than fully dissolved form, unlike nitrate. At the bottom water, that percent goes about half, but still we can say about 50% of phosphorus adds particulate. The remaining DOP, organic phosphorus, and DIP most often share half and half of the remaining. If I show a genetic cartoon of 3 layers, atmospheric, water column, and sediment process, as it's shown, are series of processes. One process I want to highlight is how organic phosphorus transform into inorganic, which is called remineralization processes. So phosphorus input either from incoming water or any other atmospheric deposition, recycle within the water column, and part of that goes in the sediment. Once it goes in the sediment, a series of processes, and some of the processes also bring phosphate into the water column. And that's one of the questions that we seek to identify the form of and the pathway of phosphorus coming back effluxes into the water column. Now, let me talk about the water column processes on particulate matter. Here I'm showing 3 sites, top 1.1, middle 3.3, and the lower one 5.2. Just the number says 1 being top and the 5 being the low. And here 3 figure shows the water column depth, which varies from about 5 meters to all the way 32, 33 meters. And as you can see, dissolved oxygen concentration in summer month, especially July, in about 15 to 20 meters, the DO goes back to almost zero. And you have a opposite relationship with the phosphate. Once the DO goes down, you have a increased phosphorus. This is a common process that there's a opposite relationship with the phosphate and DO. Some of the animation showing how phosphate changes over time. For example, March, you have almost flat line. May, the phosphate decreases towards the bottom water, and by July, it just increased significantly. For the water column particulate, and the right, I'm comparing with the sediment column phosphorus. So total P on water column, if you look at the micromole per liter, it's about 1, but micromole per gram of material under particulate phosphorus is about 100, very rich. If you look at the sediment, it is about 10 or even low. So that means the majority of this particulate on this process and water column release phosphorus or become phosphorus poor by the process they go down. That means the particulate matter in the water column are more reactive than is normally thought. One of the methods we use for inorganic as well as organic phosphorus characterization is NMR. And in NMR, we can separate a different phosphorus very nicely through well-resolved chemical sift. Orthophosphate shows at about 6+ ppm sift. Monoester has a region, diester, pyrophosphate, and polyphosphate is a very separate at the high -18 to -21 ppm sift. Let's look at the data of distribution of organic phosphorus group. In a water column, monoester and diester are the major form of organic compounds, and the monoester dominate. About 40% are monoester and 20% are diester. If we go down in a water column, that percent decreases significantly for diester, and which becomes almost zero in the water column depth. And monoesters decrease by about half. And once they settle in the sediment, you can see on the right side the phosphorus NMR of different depth in the water column. The data on the left is calculated from that NMR data. If we compare sediment at the same time, the monoester is about 20%, as you can see, very similar to what at the bottom of water column. But if you go down in the sediment, the monoester decreased by half. Diester is almost absent, even at the very top of the sediment, and the bottom I normally don't have. So definitely it shows diester degrade more quickly than monoester, which is very well known in general chemistry. We also do two-dimensional NMR, which is a proton and phosphorus NMR here, to know what particular organic phosphorus species are present and how do they change over time. Couple of compounds that is known very well is glucose 1-phosphate, phosphocholine, glycerophosphate, glucose 1-phosphate, and AMP in different time. I'm not going to go detail about how the dynamics of this different organic phosphorus vary, but we see some consistent trend, and that trend can be related perhaps to identify the microbial processes of chewing or preferring particular type of organic phosphorus. What is interesting here is that I showed you a significant amount of organic phosphorus is hydrolyzed during water column. Then question comes, where does that hydrolyzed organic phosphorus, which becomes inorganic, goes on. Remains as a dissolved phosphorus or could precipitate as some orthogenic mineral like apatite. Here I'm showing the microprobe image that shows elemental concentration in a small particle. The question is this particle detrital that's coming from the land or actually form in the water column? That question we are not very sure, but looking at the isotope value of these particular particulate matter, we are almost certain these are phosphate actually precipitated in water column. So let me move on the dissolved phosphate as well. The dissolved phosphate, if you measure the isotope value of dissolved phosphate and comparing that with the equilibrium isotope value, we can calculate how fast the phosphate is cycled in the water column. And those particular phosphorus that do not recycle also help track the sources where they are coming from. So let me touch upon the sediment now. Our question in sediment is whether the cycling is iron and phosphorus coupling. That's a classical form, well-known form of coupling, or carbon and phosphorus coupling. For this, we extract phosphorus pool from sediment, and top left, you see those pool as a function of depth. Again, these three sides, and middle side has the highest concentration of phosphorus pool. On the right, I'm showing an MR of organic phosphorus as well as inorganic orthophosphate, though, in the sediment column. Going down, here is the depth. As you can see, the top layer, 0 to 1 centimeter, can see a series of organic phosphorus, but going down, those phosphorus gradually disappear and primarily remain is inorganic orthophosphate. So we talked about the water column process before, that significant hydrolysis of this organic phosphorus do occur, but whatever goes in the sediment water interface and then gradually undergoes sedimentation, there is an active hydrolysis of this organic phosphorus as well. By extracting the different phosphorus pool, I'm talking about inorganic only here, we can find out what phosphorus pool are there and what mineral they are attached with. This is asynchronous data, quite a busy slide. I'm not going to talk about, but using iron accepts data as well as solid state NMR on the left, we are almost certain that the apatite and iron-bound phosphorus is one of the major phosphorus pool in the sediment, which is very similar what the sequential extraction data shows on the top left here. I'm gonna go quickly on this Motzbauer, just say using Motzbauer, we're able to see some iron phosphorus pool, iron pool that somehow are recalcitrant or do not reduce even if the reducing condition, anoxic condition prevail in the sediment, which was quite unique, normally not expected. So I'm showing the isotope value of two inorganic phosphorus pool, the iron oxide bound phosphorus and orthogenic phosphorus. In the right figure, those phosphorus pool isotope value are shaded. The iron oxide bound pool are called occluded or co-precipitated phosphorus pool or iron oxide, and they might also form in the water column and sediment, but they could come from the land and remain as untouched pool. Untouched means not microbially cycle in my context. The left one is the orthogenic phosphorus pool. This is phosphorus precipitated as an apatite in the sediment. The the very distinct isotope for this orthogenic phosphate pool, about 12 to 15 per ml, which is so unique and so distinct than the other pool, as well as the equilibrium phosphorus, equilibrium isotope value, which is shown as a dotted line and lit up wiggly zone at the center, does mean, and that's the only way to form, is through active hydrolysis of organic phosphorus, and the solubility of dissolved phosphate is so low. I'm sorry, let me clarify that. The solubility of apatite is so low, having a very low concentration of phosphate promotes the precipitation of apatite. So apatite is formed from the phosphorus derived through hydrolysis of organic phosphorus. So in summary, the regenerated P, that means that came from hydrolysis of organic debris settling in the water column and going into the sediment, partly triggers the immediate precipitation of apatite, and that's the only way this range of isotope is imprinted. And anything that does not precipitate could easily diffuse up in the bottom water, just following the diffusion law. So time is running, but I'm just going to show how the process works in the water column and sediment through the data we obtain from NMR. So if we were to differentiate different phosphorus, inorganic, organic, inorganic, different composition in the water column and in the sediment, we can see majority of this organic are hydrolyzed to significant extent, except monoester, which goes about half of what it is, and that process releases inorganic P in the water column as well as in the sediment column. So let me summarize these findings. Particulate phosphorus constitute a seizable fraction, a good fraction of P in the water column, and they are biologically more reactive than commonly thought during transport and settling process. Organic phosphorus remineralization is the dominant pathway of phosphorus cycling in Chesapeake Bay, both in water column and the sediment. So that means the carbon and phosphorus coupling is more strong, which perhaps is more of a new paradigm in coastal phosphorus cycling or environment where carbon is rich, a more eutrophic environment. So just keep in mind the iron phosphorus coupling are primarily seen in more of a marine sediment very strongly. The situation in phosphorus-rich environment could be different, and so is the coastal water. Those remineralized phosphorus in the sediment as well as in water column could feed the processes, microbial processes in dead zone, and continue to feed the organism for eutrophication in primarily late stage. So the question in terms of water quality is this: how those processes could be accounted, incorporated into water quality model so that management could be effective to achieve the goal. With that being said, I'd like to acknowledge my funding, and majority of work is done by the two people, graduate students, Sunanda JC and Jing Lee postdoc, right top and bottom in this slide. And I thank you everyone listening my slide. And that's the end. Thank you.
Okay, Deb. Mirko, are you there?
Hey, hello. Thank you. Thank you, Deb. Thank you. I have one question from from Andy. Please wait a minute. The question is, Deb, how do you resolve extracellular diester from intracellular diester? For example, DNA within cells.
That's a good question. For chemical method we use, we cannot separate which diester and monoester we're talking about. So what happens is the separation process. During separation process, we can separate the bacteria, bigger size, then just take the water out. We can have the cellular versus outcellular diester. That's doable, but here in this study, we did not separate those.
Okay, thank you. I have had another question. I had a question about your conclusion. You said the phosphorus is relevant for the standard quality, and also you show a graph the standard quality didn't increase in the last year in Chesapeake Bay. Do you have meeting or coordinate meeting with the government, American government, how to incorporate phosphorus or this phosphorus cycling in the standard quality? You had that kind of meeting? Because we also had to the same, we can do it our science, but the science had to transfer to the government quality like that. That's from generation new strategies, new measure to decrease the contamination. Okay, for general public, for the people. In the case USA, can you explain how to use science connect with the government or standard quality commission of the water in Chesapeake Bay, please?
That's an excellent question, because end of the day, our work like to be considered for water quality management program. Chesapeake Bay program is a very well-known and established program. It has a series of federal government body as well as a science. Partly, yes. So we are at least talking with them, and some of the senators, U.S. Senate members, reach us sometime and bring our data to that forum. I participated few of those meetings as well. So in short, yes, that could be strengthened, but there is a connection, and the Chesapeake Bay program is aware, knows this about how phosphorus is being refueled from the remineralization process.
Okay, thank you very much. I appreciate. Okay, we please keep here connected. We continue in the end of after the whole presentation. We pass to the next speaker. Please, Marco, go ahead.
So our next speakers is Miss Yanan Wang from Beijing National University from China. Welcome, Miss, to our webinar. She is presenting the explanation that is titled Assessing the Safe Operational Space of Aquatic Microphyte Biomass to Control the Terrestrialization of Grass-type Shallow Lake in China. Please, you have 25 minutes for your presentation, and after that, 5 minutes for questions.
Okay, thank you. I will start the screen. This is my slide here. This is my slide here. Yes. Because I can't see the format here. Thank you for giving me the opportunity to attend this webinar. Please allow me to introduce myself first. I'm Yanan Wang, a PhD student in Professor Bacterial in Beijing University. Today, I would like to present some work we have done before in the Bayangdian Lake. is about assessing the safe operating space of aquatic microphyte biomass to control the terrestrialization of a grass-type shallow lake in China. It will cover the following aspects: background, materials and methods, main resources, and conclusions. Lake is an important wetland ecosystem, and for China, it is 83,821 square kilometers and accounts for 19% of the total wetlands in 2015. And it provides a range of ecosystem services, such as supplying water resources, controlling floods, regulating the regional climate, and maintaining the regional ecological balance. Well, in recent 20 years, 72% of lakes faced different stages of lake shrinking or disappearance, and has become the main problem for the shallow lakes. Therefore, more attention should be paid attention to it. However, for now, there are few studies about it. Well, what is terrestrialization? It is the succession process from lake to a terrestrial ecosystem due to the accumulation of sediments and detritus deposition in the lake bottom. And there are three driving processes for it: the physical processes, the chemical processes, and the biological processes. We know that physical process is the basic driving for it, especially for the hydrologic process. It is the basic for the wetland formation, development, and evolution. And the sand deposition leads to lower water depths, providing a precipitator for aquatic plant spread. While for the chemical processes, note that the biogeochemical cycles of elements such as carbon, nitrogen, and phosphorus, the absorption, accumulation, distribution, and return among the components of the ecosystem, they all affect aquatic plant growth. Well, for the biological process, we know that the macrophytes grow and die, and the lakes are gradually filled with sediments and plant detritus. So, aquatic macrophytes are considerable reliable indicators for detecting lake status, and their deposition is a key factor for lake terrestrializations. So, I will turn to the biological deposition. As I said before, the lakes are gradually filled with sediments and plant detritus, and then it will result in lower water depths, and the macrophytes spread from the littoral zone to the center. We know that the growth and spread of macrophytes highly depends on lake sediments and the nutrients abundance in the water. And the sediments create a favorable habitat for macrophytes growth. In turn, macrophytes can also embolize sediments and promote its deposition. And then the lake sediment layer continues to rise due to the high plant detritus input, which stimulated overgrowth of macrophytes. From the picture, we can see that the macrophytes increase in the macrophytes biomass and the decreased water level, so they can damage aquatic life habitats and accelerate the lake terrestrializations. How can we do that? There are several manners we have done, such as the ecology for water replenishment, ecological dredging, and mechanical harvest. Well, for the ecological water replenishment, in China, the famous project, the source-to-north water diversion, has successfully implemented, and it has avoided the risk that the water level reduces or sediment dry. However, it also creates an inflow processes and influences the abundance. Well, ecological planning and maintenance harvest will always use the method to reduce accumulation of organic matter, and this method can effectively avoid accumulation as well as create nutrient loading. Now, there is a complexity of the source loaded into the method, and the spatial distribution can hardly on the macro side what it is matter. Now, I will introduce the next behavior. Apart the macro side and control, it has been proved to making cost-effective and environmental design approach, and they have used the chemistry project. From the picture, you can see that before control it is in the heavily terrestrialization stage, and after control, everything got better. The biomass has decreased, the water level increased, and the water quality has been better than before. Because we define the safe operating space as a reasonable threshold range for each plant biomass. And these methods can optimize the spatial distribution patterns of the macrophytes with the proper timing, scale, and cutting amount. And I will show the method in details. We use the method in the Biennial lake. This study area is also the population program. And this lake is 115 kilometers away from Beijing, our capital. And this lake is a key part of the Xiong'an New Area. It's a national level economic development zone in China, and it is of great strategic significance for China. This lake is the largest grass-type shallow lake in the North China Plain, and it enjoys the reputation the kidney of North China and the pill of the North China. While this lake is with high nutrients, high microphyte density, and lower water depth. What causes it? The anthropogenic disturbs such as aquaculture, wetland tourism, sewage discharge, and upstream decrease. This all greatly undermine the self-management capacity of this lake, and resulting in the spread of lake macrophytes. And for during the past 33 years, we can see from the figure that the water surface had decreased 26%, and the swamp had decreased 19%. All this has severely aggressive terrestrialization. Okay, to concept our method, I put a technical frame here. And the step one is for the community investigation. We use the field investigation to find the domain plans and the communication patterns here. And we got the data here, and we use all the data to determine the safe operating space of the macrophytes biomass. Okay, for step one, the community investigation, we choose 6 sampling sites. According to previous studies, the white table coverage and water quality indicators were applied to define the stage of the terrestrializations. So the Captain, Duancun, Zaolingzhuang were at the lightly terrestrialization stage, and Quantou and Wangjiazhai were at moderately. Only Captain were at heavily. And thus, the donating macrophytes here, including 3 emergent plants and 2 submerged plants here. And for the natural community, we found the 2 signal community, the first married Australia and the fatal Muscali Tapinatus. And to To be simplified, we use community 1 and 2 for further study, and the 3 mixed community here for the community 3, 4, and 5. And then we use the harvest method, field investigation, lead back method, and sentiment analysis to gather data. Here I want to anticipate the lead back method. We know that the classification 1 is to determine the deposition rate, and we combine it with equation 2 to get a relative deposition coefficient AI for the average population, and then the sentiment analysis. As we know, the surface sentiment main conception of plant properties. So we use the plant property density and equals to the sentiment to the back density matrix by the content of the soil organic matter. And after we got all the data, how can we determine the state of this space? For microsite format, firstly, assuming that the rate is complete transformation of the area, and we use the average depth of the rate and the maximum depth for less calculation. And the value we use here is the first grade meter. And we can use the equation 4 and 5 that m average depth utilization rate. And then they can use the equation 6 to draw the total energy amount of microsize and P. After we go that, according to our we have got the utilization rate AI and the total rate the total dry rate utilization and P. So we can use equation 7 to get the dry rate utilization for each population. And then according we have got the MTI and the K value, or the value we can substitute to equation 1 to get the annual deposition of our macrophytes. Now we can determine, we can assume that the lake will be completed terrestrialization after N1 to N2 years. And now we can determine the safe operating space for it. The biomass for S2 is P2 to P1. Okay, I will show some results for my study. First is the plant biomass at the community level. We have calculated the biomass in May and September. From figure, we can see that the plant biomass peaked in September, and the accumulated biomass generally increased compared with that in May. And what's more, the biomass of community 1, 4, and 5 was higher than that of community 2 and 3. And these 3 communities were dominant macrophytes in aggravating the terrestrialization. Well, what's more, in the early stage of rapid plant growth in May, terrestrialization exhibit less impact on plant biomass. The community biomass was deeply influenced by terrestrialization through plant growth, especially the biomass in the community 1 and 5 in September. Well, for the plant biomass at the population level, we can see that the terrestrialization mainly affected by changing habitat factors, inter- or intra-specific competitions, and the single species communities were more conducive to plant growth than the mixed communities. And Damasomes was more sensitive to terrestrialization in May, and the Australia's single species communities respond strongly to terrestrializations in September. And now we got the biomass we can use substitute all the biomass into the equation we have mentioned before. And the measured years we set here is according to the model we have used before. So the measured years for the likely calculation we set is 115 to 118, and for the moderation we set it to 100 to 120 years, and for the family we set it to 15 to 17 years. So with the managed years, we call the cell deposition rate here. Other side, we can use that to get the deposition rate and the relative deposition coefficient for all the population. And for the single community, the relative deposition coefficient is equal to 1, and for the mixed community, the population, the sum of the population for all the relative deposition coefficient is 1. And here we go. We got the annual accumulation biomass for the population, and the same for it. We can get the safe operating space for the community. And now we should compare the current biomass to the safe operating space. And we can know that the higher the current biomass are, and the more severe the terrestrialization is, indicating there is more pressure to management the terrestrialization lakes. And generally, the current biomass values were higher than the target values, especially in September. And it is deserved to mention is that the current biomass in May is lower than that in September, while the safe operating space in May is higher than that in September. It's meaning that management pressure was higher in September in those zones. And at the communication levels, we can say that for communication 1 and 4, the current values were much higher than the target values, indicating that the management pressure is very high in these communications. While for communication 2 and 3, the values were very similar, suggesting that management of this communication would be easier. And now we can draw the conclusions here. We have estimated the safe operating space of microphase biomass in different terrestrialization stages. And I think this method supplies analysis for reducing domain macrophase and controlling lake terrestrializations with proper timing, scale, and quality amount. And we have published this work on the Journal of Environment Management. If you are interested in lake terrestrialization, you can download it in the below. And for future work, we should consider the influence of water level, the hydrodynamics, and the water quality on the plant biomass to optimize this method. When we want to control the algae tablet, what we should do, how we can optimize this method. Maybe we will consider this later. Okay, that's all. Thank you for your attention.
Thank you. Thank you, Yanan. Please, Marco over there. Are you there? Marco?
Yes, thank you again, Janan, for your nice presentation. Very interesting, Sam. We have 2 questions for you. First one, if you know, if terrestrialization affect the aquatic plant microbiomes interaction, influencing, for example, the growth or tolerance of macrophytes. Do you have information about that?
Yeah, that's a good question, and we will consider it later in this presently I just use the biomass to calculate the safe operating space. Okay.
Maybe for next steps.
Maybe further,
No?
yeah.
Yes. This is the future.
Yeah.
The other one question for you is how we save operational stage quantified? What ecological parameters used to calculate different years? And if this is only for microphyte?
Yes, the safe operating stage here is the threat range for not a threat. And we calculate the different managed years, use the model we have done before, and we can go the safe operating stage. And this is now we can use it in the for the macrophytes biomass only. Yeah, maybe further we will use for combine it with plant macrophytes in action.
Okay, I have the last question. It's very shortly. If you, how we know plants can acquire nitrates like phosphorus. Thinking in this, I think that there may be a bigger biomass of microphytes could be better to the phosphorus acquisition, but could be the opposite one. Do you know if it is better to have more microphytes or control this?
Yeah, we use the demonstration project. Use the scale for it is just for 10 hectares, so it's just a small scale. And maybe further, we will use on a large scale for it.
Okay, thanks very much.
Okay, thank you.
We don't have more questions.
Thank you. We move to the last presentation. This presentation I have to introduce to Marco Campo. Marco had a career in environmental microbiology, and we start to this topic about aquatic microbiology in our laboratory. But Marco now he will present the data about the last sampling. Marco is working in the river and lake. You know, it's this area where we live in Chile, and the south of Chile had a lot of lake and river. That's very relevant for the people, for the economy, for the culture, for the tourists. That's now we are going to introduce the study about the bacteria involving phosphorus and other nutrients. Marco had more data about that, but now it's focused in phosphorus. They share with us the presentation with the data collected from this area in Chile. Please, Marco, go ahead.
Okay. I'm sharing the slides. Can you see this? Okay. Are you also hearing me? Yes. Okay. So I'll start my presentation. Sorry. Okay. I'm glad to be here in this first microbiology and webinar from our laboratory, and I'm very glad to share with all of these inviters and share my part of my postdoctoral investigation. This work is entitled as an approach to the spatial temporal distribution of phosphorus content, phosphomonosterase activity, and bacterial community in sediments of two rivers from southern Chile. As we know, and we are speaking about, phosphorus acts as a limiting nutrient for microbial growth in aquatic ecosystem, like rivers. But there, large supplies of this nutrient can lead to the blooms of faster growing microorganisms, such as phytoplankton, leading a concerning process called eutrophication. This status can remain by large periods of times because phosphorus is able to change between inorganic phosphorus forms, Pa, and organic phosphorus forms, Po. and accumulate there in the sediment of these systems. From this, PO accounts close to the 25 to 75% of the total phosphorus in sediments, being molecules such as phosphodiester and phosphomonoester, the main forms that we can find there in sediments. In sediments, PO is complex with the unstabilized with metal ions and organic matter, but the spatial-temporal fluctuation, the physicochemical properties can transform on this organic form into PA forms more available for microbial use and lead again to the eutrophication process. There in sediments, bacteria can participate directly by the release of some phosphomonosterase enzymes, PASE, okay? From here, I call this like PASE. There are two isoforms, the alkaline phosphomonosterase and the acidic phosphomonosterase seems to be key process in this phosphorus recycling. These enzymes are widespread in the bacterial community and has been interestingly associated with eutrophication. So in recent years, we have having a lot of investigation in this area. For ALP, two encoding genes, the 4D and 4X genes, as well as for ACP, the 4C gene, has been well correlated with the PASE activity, but poorly understood for their ecological roles in these systems. For these reasons, studies focus on reveal the effect of phosphorus in the microbial community and PASE activity using advanced tools such as the whole-truth sequencing analysis are required. Here, we have two rivers, two important rivers. These are Imperial River and Torten River, and two water bodies supporting the economic activities of our region, the Araucanía region in southern Chile. Their catchment has been historically under intensive use of anthropogenic pressures, which have increased the phosphorus input into their water bodies. However, contrary to the common belief, this P enrichment has not been associated with documented eutrophic episode in the last years yet. Thus, as there are not existing studies focused on the microbial community and their ecological role in recycling, the aims of this study were, first, to determine the P status of sediments, to analyze the PASE activity and PASE bacterial gene abundance, and to reveal the major factor moderating the composition of bacterial community. The material and method of this study were firstly for the sampling, we obtained samples from 4 sampling points of from every river, and these were separated as proximal ones according to the closeness to the Andes Hill, or distal ones in order to the closeness to the ocean. Okay, these sampling points were Nueva Imperial, Carahue, Tranapuente, and Nehuentude from Imperial River, and Villarrica, Pitrufquén, Nueva Tolten, and La Barra from Tolten River. From every sampling point, triplicate superficial sediments were collected in winter 2018 and summer 2019 using a Petersen dredge. And this conform is the temporal dimension of this study. At the same time, every sampling point was measured for some parameters such as temperature, conductivity, dissolved oxygen, and pH. One time we obtained the samples, we deposit the sediments in plastic bottles, and we transport this immediately to the EMA lab for the PASE activity measurement, DNA extraction, and later chemical analysis. For the total carbon and nitrogen, we use elemental analyzer. And for the estimation of a total PA and total PO, we obtain this after a sequential extraction using different extractant solution, as this is shown in the table. For the PS activity, we use paranitrophenil phosphate as substrate, and after DNA extraction, we use this DNA aliquots for the real-time PCR estimation of the functional genes for C, for D, and for X, and as well the total bacteria by assessing the 16S rRNA gene. And we use this to relativize the functional genes. After that, with all of this information, we construct a principal component analysis to see the distribution of samples. According to the PCR results, we select some interesting samples for the high-throughput sequencing analysis. And we amplify this, we index these samples, and we sequence this in the Illumina MySeq platform available in BioRen from our university. The obtaining reads were later processed, and with this information and the above information, we construct a redundancy analysis to see the parameters with the most important factor modeling the bacterial community. Additionally, we carry out the RichNet, the alpha and beta diversity, the taxonomic assignment, and concurrence networks. Also, parametric tests by ANOVA and Pearson correlations. The results and discussion of this investigation. Firstly, in general terms, the spatial temporal difference among physicochemical properties and phosphorus content were statistically different in all of these parameters. The most remarkable difference were between PA and PO, being PO sometimes threefold higher than PA in several samples. At the spatial dimension, P was mostly higher in the distal samples, these samples close to the sea. And this could be related maybe about the finer texture of this sediment, due that this texture is more, these sediments are easier to fix more nutrient like phosphorus. The temporal plane, the difference were more notorious, okay, being the highest value found in the Warner season for summer. Overall, the PO fraction, the organic fraction of phosphorus, which were also higher in summer. And this could be related with the accumulation of detritus of aquatic dead biomass, and maybe by urban influence by wastewater inputs. Additionally, temperature and DO were correlated because when it's increasing the temperature, DO is commonly to decrease because microbes are increasing the activity and use more oxygen. And also, as in summer, the water decrease, conductivity can increase also. In the case of PACE genes and PACE activity, comparing both isoforms, we found the higher activity for the acid isoform. Here, the spatial difference were also significant for the distant samples. However, again, the temporal difference were most consistent and peaked for the summer again, establishing a possible relationship with the higher PO values, as I told before. The capability, as this PO is capable to stimulate this activity in eutrophic waters. The distribution of these genes positioned the FOC, the acidic isoform, as the most abundant gene in this environment, followed by FOD and FOX, showing a similar trend and spatial temporal distribution like the above parameters. Regarding FOD and FOX results, authors say that the existing studies say that the FOX could be higher than FOD or conversely FOD than FOX, being both genes widespread in this kind of environments. However, regarding to the FOC results, no existing studies until the date are available describing the abundance of this gene in this kind of environments, which becomes or results as usable information to understanding the P genes relation in this kind of systems. According to the principal component analysis, this explains more than the 60% of the variability of the samples and parameters. And this did a clear separation of samples according to their river provenance and the seasonal provenance, clustering them in different quadrants. Samples showing tightly positive orientation with base activity and P content were mainly from summer. On the other hand, the opposite, having the most negative correlation, were from winter. Similarly, base genes were able to locate the most distal sample in different quadrants, being the summer here in this quadrant and oppositely the winter ones. Accordingly, the Pearson correlation test among all these P related parameters show lot or several positive correlations, and almost all of these were significantly, being negligible the negative interactions. And these correlation could present direct evidence about the stimulation of overloads of P on the base activity genes with a particular behavior with the PO stimulation. Finally, based on the PCA results, we select Nova Imperial and Trena Puente from Imperial River and Petrofegen and La Barra from Tolten River for the next study by the Haupt sequencing analysis. There, the highest richest values were for the distal samples, but again, the temporal difference were strongest, with the temporal difference in the highest value were for the colder season. Similarly, Shannon index showed a different result as the spatio-temporal with peaks for winter. The beta diversity did not show a spatial difference, but distribution was strongly influenced by seasons. Then the spatial difference could be related with the sediment texture and maybe by the nutrient content, but controversial this result because the ecological heterogeneity of a river is high. Similarly, the temporal difference could be related with the trophic increment during the transition from the colder season to the warmer season, but also controversial because when rivers has a very high polluted level, even no seasonal change could be observed. For the taxonomic assignment, we observe the main difference at the temporal dimension, being Protobacteria and Bacteroides, the higher phyla during summer, and Actinobacteria and Planktomyces, the most abundant in winter. Therefore, Protobacteria and Bacteroidetes have been indicated of major abundant phyla in environment under human impact and eutrophic environments. Also having a lot of groups with the harboring bacteria, whereas Actinobacteria and Planktomycetes have been reported at pure mineralizers bacteria, overall during algae blooms. At the family level, Hypomicrobiaceae, Chitinophagaceae, Pirulaceae, and Gammataceae were the family most abundant during winter. Otherwise, Camomonadaceae, Cyanobacteracea, the Sulfobacteracea, Chromataceae, and the Sulfobulbacea were for summer. Several of these groups play ecological roles in the recycling of phosphorus in aquatic environment, being some of these able to couple the phosphorus mineralization with the intracellular sequestration of phosphate in molecules such as polyphosphate, poly-P, that is an important molecule for starvation of phosphorus. The redundancy analysis show that in the RDA in this plot that the community and environment relationship were best explained by PA, LP, PO, ACP, and FOC and FOD. This showed nearly hilled spatial temporal separation for bacterial community. However, there's apparition of some isolated bacteria there in the plot from the total community, and their relationship with the sample were observed. These results suggest that the FOC, FOD, PO, and ACP as potential factors with roles as predictors for the variation of bacterial community. Then, for example, the Sulfobacteracea, Chromataceae, and Conomomodaceae families have been described as active couples of sulfur oxidase reduction with the organic matter respiration and metal oxide reduction, a process able to increase the release of dissolved phosphorus and able to stimulate the pH activity. For the co-occurrence networks, the analysis showed a higher topological value for the winter, okay, which revealed the higher niche differentiation of this network bacteria. This is a bacteria-bacteria interaction, okay, and revealed higher niche differentiation, higher network complexity, and lower ecological pressure than the summer, because this network was smaller. According to our analysis, 13 keystone taxa were identified for the winter network, and just one was exclusive for the summer network, and 3 were shared between both networks, being this Chromataceae, the Sulfobacteracea, and the Sulfobulbacea. Here, the existence of this shared core taxa could be a reason to demonstrating the role of this taxa as buffer or stabilizing taxa between both networks. Also, these taxa present almost all or completely positive interaction, again supporting this central role in stabilizing the bacterial community. Overall, during the stressful condition that is summer. In this context, our results becomes valuable information about the contribution of sediment bacterial community possible in order the trophic shift of Bob River, as we saw in the previous slides. Nevertheless, deeper studies linking specific molecular indicators involving the phosphorus turnover and keystone taxa are required for a better understanding of this phenomenon. The conclusion and outlooks of this study, so we can first to say that the P was the most unbalancing physicochemical parameter at the spatial plane, but mostly at the temporal dimension. PO fraction was the largest fraction and was correlated with the paste activity and paste gene abundance, describing a suitable stimulation of the paste harboring bacteria population. The composition of the total bacteria community was notably less diverse during summer, being P-related variables possible responsible of these changes or shifts. Network shift ratified that the summer was the most stressing condition, and the existence of some core taxa such as Chromatiaceae, the Sulfobacteriaceae, and the Sulfovolvaceae probably reveals the role of these taxa as buffer in the stabilization of community. Thus, given the relationship between PO and the spatio-temporal shift of cement bacteria, the present study represents valuable information approaching the understanding of this interaction in the pericycling in eutrophic freshwater ecosystems. I want to acknowledgement to my entire team, the MLab team, also for the Fondesit and PCA program that supports this presentation or also this webinar. To everyone that have heard me in this moment. Thanks very much, some reference, and thanks very much to everyone.
Thank you, Marco. Okay, I had a question for you from Dave, JC. The question is, are the distinct difference in summer and winter caused entirely for natural ecological reason or some anthropogenic contribution as well?
The results there, the literature is not contundent with this because in natural ecosystems, sometimes the shift in the seasonal shift can manage this the structure of bacteria, but sometimes it's not changing this. Also, in environments heavily impacted by a human, can happen the same. So this depends of the season and depend of the heterogeneity, for example, of rivers. That they are a lot of ecological niche.
Okay, I had another question from Andy. Marco, very interesting work. Have you published it yet?
Where this result will be part of the next article.
Yes, we are writing the article that we hope to publish soon. We had another question from Ron Xiao. Phosphatase harboring bacteria. How to know the bacteria is phosphatase harboring?
Just because we assess this by the genes. We don't know if they are expressing the genes, but at least this population or bacterial community has the genes, these 3 base genes.
Yeah. Okay, I have a question, Marco. Is you see the last presentation from Andy? Andy showed the abundance of phosphatase gene in Everglades. Okay, and when I check your data, I think your phosphatase, the abundance is lower compared to Everglades. My question is, do you know how is the abundance in different ecosystems, aquatic ecosystems, for example, rivers, in your case, estuarine, in the case of Everglades, and bay, seawater, coastal bay in the case of J? What is the abundance? Is similar? Because I think your data is show very low abundance compared to...
Are lower, yes. We have also results from other systems because you have to separate the lotic ecosystems, that the rivers, streams, the lentic ecosystem that are lagoons, lakes. The dynamic of this environment are different because, for example, rivers, they are prone to be to the scavenging of the water flows. So the deposition of nutrients there are different. For this reason, we did the separation according to the proximal or distant locations, because it is more probably that in the distant location to deposit more nutrients. So, and also this is very dynamic about the seasonal shift. So I think that in Everglades, this is a lentic ecosystem, so they should to be more abundance because there are more deposition of nutrients and the process are more quiet, not too dynamic than a river.
Okay, thank you. We pass to the next stage in this seminar. We invite to all speaker. Please can open the screen for show the all speaker is present in this meeting. And we also invite to the audience to make some questions. Or we already had some questions from the audience or from the speaker, and we need to open. Please wait, Andy and everybody there. Okay, please wait a few minutes for introduce all people in the screen.
I think we're there.
Okay.
Okay.
Yeah.
Hello. Also, I think I want to welcome to Professor Bai and also Cho or Xiao. Please also we wait because we start to collaboration, with Chinese with the Beijing Normal University. We had a new project collaboration, like a project similar to Deb and Andy. We had both project. It focuses in the aquatic environment in the South Chile. Okay, bacteria involved in nitrogen cycling and bacteria involved in the phosphorus. Okay, we said thank you to everyone to join us with us to this meeting, and we will start to round table based on the question and comment made for the audience. I have to hear some question for Andrew. Is made by Rodrigo Vera. The question is, how can they increase the atmospheric CO2 concentration change the community? About your presentation.
Yeah, I can you hear me? I think I should be on, right? Honestly, I don't think that there's going to be much change in our system with increasing carbon dioxide concentrations. The reason for that is that in general, you think that CO2 is not limiting in aquatic systems and sediment-dominated systems because there's so much carbon dioxide that's produced. And that's particularly true in our system because it's carbonate-based. It's underlain by by calcium carbonate that came from coral reefs. And so there's just a lot of carbonate and a lot of carbon dioxide in those systems. So I don't think it's going to have any impact on us. There may be systems that are different that are limited in carbon dioxide, but I'm not familiar with those.
Okay, thank you. I had another question for Deb. Deb, this question made for Andy. And I think I really sorry. I had another for Professor Bai. Hi Deb, I want to know how did you identify the remineralizer phosphorus in water and the phosphorus released from the sediment? Still, I think the salinity will be greatly influenced by phosphorus mineralization.
That's a good question. So the salinity aspect was not included in my presentation, even though salinity has significant impact on phosphorus speciation, and given the microbial composition vary as well, that will impact the phosphorus mineralization. So those three sites, the top one, salinity is about 1, and the bottom one has salinity could go as high as 28 to even 30, very close to the ocean environment. The research here is only on the vertical processes, like settling processes and remineralization. The first question was on how do I quantify remineralization. Is that correct, Nilko?
Yeah.
Okay, so two ways we quantify. First, just to measure the concentration of this organic phosphorus, and both by quantifying organic phosphorus as well as quantifying inorganic, and do a mass balance analysis. That's the one way. The other way is using isotope, because we know when this particular organic phosphorus break down, they have a one particular isotope value. Since we know the composition of all organic and inorganic, we can empirically calculate what would be the isotope if those those organic molecules are hydrolyzed. And sometime it's in this study, the inorganic phosphorus was precipitated right after hydrolysis, and that allows us to identify the processes.
Okay, thank you. Now, I have another question from Xiao. But we have Ron is here. That please make your question, please.
I want to know what is the indication of your findings for the land or the soil management for the government or for the manager?
Oh, a big question for each one of us.
Yeah, you have very interesting results and different findings and different results, many data. So just give some examples of the uses of your findings. Can you? Yeah, the government always ask the scientists to use useful the data useful for their management.
What the practical about your data?
Yes.
We can use it for something.
Yes.
Not only of course, it's very important for the paper, but when we have to talk with the government. There's a similar question. We have to office the people. You always talk with the government officer. How to
for example, the project we will study in the Baiyangdian Lake. We check the gene. We check the antibacterial gene in the water. So after that, after we have some data, we get the results, and we will build a free guideline for the water quality management. So how to do that?
For example, in the case of us, with this project that we are studying Villarrica Lake, we are working with the ministers, with the representative of ministers. So, for example, for the first sampling, we did the sampling with the people that is doing the sampling at the same time from the minister studies. We are trying to couple all information with the information that is available from the minister, environmental minister. So what is the idea of us is try to couple this information, the molecular information, that could be faster than other parameters that are more to contundent, for example, try to have a approach to quick response to see, for example, about the status of water quality in Villarrica Lake, or try to predict even if there are could be in the next days, for example, or next month, algae bloom in this kind of environment. We are just starting. This is a lot of work, because we have different language with the ministers, with this kind of organizations. So it's also new for us.
I think I could also answer Xiaorong's question too.
Thank you.
What we're doing in the Everglades is more descriptive. We're just saying what these management effects have had, and they know what to do. They know to stop the input of phosphorus. But part of the story that I haven't told you is mercury cycling, that I think is also of great interest in China. And
Yes.
what we're thinking is that based on what we have, that water management might be critical in suppressing mercury or controlling mercury methylation. That's important. So that's a management issue that's come out of our work. So one of the things that they do in the Everglades is control the amount of water that flows in at different times of the year. And we're thinking that probably through judicious control of water, that those mercury transformations could be controlled a little bit better than they are. So, but I know that that's of interest to China. So
Yes. That's a big problem. The
Yeah.
water pollution. Yeah.
Yeah.
I always think about how to use the result or the data for the practice use to give the practical support to the public.
yeah, that's very important.
Yeah.
Yeah.
That's what we will do in our project with Miyoko, with Professor Miyoko and Professor Bai. We will find what is the result. We'll give the suggestion for the government management in that area.
Mhm.
Yes.
Yes. In this stage, we are just descriptive. What happened there? Because we don't have more information. Because we
Yeah.
have different river, different city, big city, small city,
Dynamics.
had the input from the agriculture or aquaculture, that we had a lot to do. That had to collect some information and then show to the government officer. We start discuss what we can clean up and what data can be useful for in the future. Make some strategy about that or management control. We don't, now is only focus in the physical chemical parameter, no involve biological or microbiological parameter in Chilean regulation. Yes. I think in this stage very relevant to not only us, also another group is motivate to do that kind of the work in the South Chile. That's very nice. I thank you for supporting this challenge for us.
Thank you.
No, I check here. I think not hard to question about the audience. So there's so many. Hello. There are some comments. This agriculture use big amount of nitrogen and phosphorus near to the river.
For sure.
From
For sure. Yes.
Luis, that he's the director from the BioRen. You know, BioRen is the big center here in the Bio University, and he attend to this meeting. And maybe you, Marco, you can talk about the use of the agrochemical in this area and what you think is the impact for the river and the lake in this area. Maybe you can explain.
Yes, the catchments of Tolten River and Imperial River, also Villarrica Lake, and all the small one lagoon that is Budi Lagoon, or we can say in all of our region, the soils are very erode. The agriculture use a lot of nitrogen and phosphorus-based fertilizers. Also, a lot of pesticides. And we have, for example, in the north of Villarrica Lake, a lot of aquaculture. So the inputs of fertilizers, also soil erode particles that is reaching these rivers and these lakes are increasing every year. For this reason, we have, for example, we are trying to study the antibiotic resistance in Villarrica Lake, in rivers also, if it is possible. But by the moment, we are studying how bacteria are participating in the phosphorus cycle. For example, in the Budi Lagoon, we have there also the problem with the erosion of lands. The lands there are have a tilt. With the increasing tilt, more erodes particle salts are reaching the sediment. So we are enriching every time this water body with more nitrates. We have a big problem with this, and the deforestation also is a big problem for us.
Okay, thank you. I have another question for you, Marco. Sorry.
Okay.
It's made for Dr. Gonzalo Gajardo from Los Lagos University.
Okay.
He said you find it two different, summer and winter. Okay, it's big different. Yes. Also, the flow, water flow is different because the winter is rainy season and summer is dry season. Do you have some data about the how many water, the flow, the water in the river? Because this, I think, or he said, is a big parameter to be considered.
Yes, I don't have the hydrological parameters, the results, but for sure the climate change is affecting also the diversity of microbial communities in sediment and also in waters, because if we know that the waters and sediments can be very different, all are correlated. So we can have if we have a more drier seasons in the next year, for sure we will have this will be affecting the community there in this kind of environments.
Okay, thank you. Do you have another question or comment? Because I have my question. Let me check again. Yes, I think I finished with the question from the audience. Okay, but if no question, I can appreciate your participation in this meeting. Thank you very much. I know you are very busy. You had a lot of work to do. Thank you very much to connect with us and share your presentation. I hope that in the near future may continue with this meeting and obtain more data to share with you about both project to us, or maybe your project also you can share with us and discuss about that. And I know if we continue contact by mail, that's no problem for continue working with you. Thank you very much. And
thank you very much also again.
Before finish, I want to thank to invited speaker, our audience for your contribution. We expect continuing strengthen our national and international collaboration in this and other projects we have in the laboratory. Also for Chilean audience, stay here there. We had an interview in national radio this evening at 6:30 p.m. about this topic that we had maybe published on the newspaper about our project, and now in the Capital Radio interview me about this project because it's a very important topic and relevant for Chile. That Chilean audience, all people invited to listen to the radio interview, maybe short interview, not so much, but I'll talk about this topic too. Okay. Finally, I want to thank to the people from the company, webinar company, CL Pro, for your technical support in this meeting. It's the first time we try to do this in this modality. Usually, you come to Chile, we have a local seminar, and you visit the lake near university area. But now we have to connect by internet. But I hope in the near future we have to continue in the next year in the collaboration. I hope you come to Chile and we prepare a presential meeting with you. But in any case, in this local digital meeting, I appreciate the collaboration, the technical support from the CL Pro company, and also the collaboration with you. I want to say, if you want to say something to finish, or we finish the connection.
I want to say thanks very much to everyone. It's good to see all faces and also new faces. And I hope to meet you in person. the next year, if it is possible. I don't know who will be with the vaccine first, but we need this to share with you and work with you.
Okay. Is nothing more? We have to say hello, goodbye, and see you soon and contact in the near future. Thank you very much for your attention, and thank you for much for all people connected with us. Thank you. Bye-bye.
Okay.
Thanks very much. Bye.
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 Bye.